Using computer animation based on molecular research, we are able to picture how DNA is replicated in living cells. You are looking at an assembly line of miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called a helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules. Transcription is the process by which DNA is copied into RNA in the first step of gene expression. It begins with a bundle of factors assembling at the start of a gene, that is, a linear sequence of DNA instructions, here shown stretching away to the left. The assembled factors include an RNA polymerase, the blue molecule. Suddenly, RNA polymerase is let go, racing along the DNA to read the gene. As it unzips the double helix, it copies one of the two strands. The yellow chain snaking out of the top is the RNA, a copy of the genetic message. The nucleotide building blocks that are used to make the RNA enter through an intake hole in the polymerase. In the active site of the enzyme, they are then matched to the DNA, nucleotide by nucleotide, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, thymine is replaced with the closely related base uracil, commonly abbreviated U. You are watching this process, called transcription, in real time. When the mRNA is complete, it snakes out of the nucleus into the cytosol. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. tRNA molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the tRNAs. There are different tRNAs for each of the 20 amino acids, each of them carrying a 3-letter nucleotide code that is matched to the mRNA in the machine. Now we come to the heart of the process. Inside the ribosome, the mRNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to the 3 corresponding letters on the tRNAs. When the right tRNA plugs in, the amino acid it carries is added to the growing protein chain. You are watching the process in real time. After a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends on what genetic message you feed in on the mRNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.